I am the chair of the AMPA National Membership Committee. Today, I will be introducing a wonderful person who I've worked with on the AMPA Membership Subcommittee, who is a huge asset to what we're doing. He is a board certified, fellowship trained orthopedic surgeon. He is a member of the Georgetown MedStar Orthopedic Institute. He is the co-founder of the Center for Advanced Specialty Surgery with 2 locations in Nigeria. He is an avid philanthropist and the founder of Operation Stand, Walk, Run, which is a nonprofit organization for helping people in developed countries stand, walk, and run. He is also a humanitarian award recipient, the chair of the AMPA DMV chapter, and a formidable force to be reckoned with. I introduce this afternoon Dr. Aham Onyike.
Thank you so much, Suzanne. Every time I hear you speak about me, I feel like I should be recording. But thanks, it's obviously very flattering. Good afternoon to everyone. Thanks again. As mentioned, I'm the chapter chair of the DMV chapter of AMPA. This is an AMPA event. I'm a co-moderator today. So welcome back, our colleagues and guests. It's not just AMPA members and it's not just physicians. We recognize that we have a wide array of participants. It's a continuation of our dialogues on COVID-19, particularly the vaccine. I think anyone who was on prior broadcasts will accept my unbiased impression that the discussions have been magnificent and certainly very informative. Today's discussion is also being streamed live on YouTube to help with some capacity challenges that we encountered last time. So we last met 3 weeks ago, and this is our 4th meeting. 3 weeks ordinarily will not sound like a lot, but really a lot has changed. I personally find it very fascinating, the continuous evolution of discussion and the flood of information on COVID-19 and everything pertinent to that. It seems like more data emerges every single day. We just heard that there's some exciting news coming out of Nigeria, and curious to hear what that is. There's of course gathering evidence on the vaccine in all facets and nuances pertaining to that particular subject. So today's discussion will be very full. Hopefully we'll keep it to 2 hours, but I'm confident we'll cover a lot of ground. As usual, we have an excellent crew to lead us in discussion. Before we get there, we can begin with some simple numbers. Here in the US, there have been close to 26 million cases of coronavirus and close to 450,000 deaths in just over a year now. So it's very staggering. For over 7 days in December and 16 days in January, we had close to 4,000 people dying every single day. And this number, unfortunately, includes many doctors, including some in the AMPA family. But thankfully, in spite of all of that, there is some optimistic news. Obviously, the vaccines have been rolling out after what will be described as an initial bumpy phase. Most will agree that the initial rollout by the prior US regime was somewhat extemporaneous. But there's a new administration now and a different approach, including a target of 1.5 million Americans being vaccinated daily. So far in the United States, 38 million have been vaccinated, so that's a lot of data. Out of that, 38 million, close to 7 million have received a second dose. And it appears that the so-called third wave in the US has started to crest. The numbers appear to be coming down, both in terms of new infections, deaths, hospitalizations, which includes intensive care unit admissions. And the vaccine hesitancy is also decreasing. So there is a lot of reason to be optimistic, but nevertheless, there remains some concerns, particularly about variants. Some like the UK variant have shown a higher transmission rate ranging between 30 and 70%, and Dr. Ofotokun could elaborate on a lot of these facts. And there is some susceptibility concerns to some of the existing vaccines, particularly with the South Africa and Brazil variants. which may offer some degree of immune escape, meaning that the vaccines may be less effective. Again, our panelists will elaborate some more. In addition to all of that, is a growing recognition of disparities in vaccine administration among minority communities here in the US. So many of our own communities and our own patients. There have been a lot of bottlenecks in this mass inoculation program. Initially, there was a problem with distribution, and now it appears to be more of a supply issue. As such, there's somewhat of a new dilemma that's creating a discussion as to what's the better approach. Should we try and get as many people one dose or get as many people the recommended 2 doses? So a lot of questions to be answered and trying to understand what's the best public policy. Another emerging hot topic is that pertaining to reopening schools. The Center for Disease Control recently put out a very strong, compelling editorial in JAMA recommending that schools be reopened. They feel that the evidence currently seems to indicate that it could be done safely, but the teachers' unions are resistant. We also have 3 emerging vaccines in addition to Moderna and Pfizer. And again, our panelists can give us some more insight into these. It includes the Oxford AstraZeneca, Novavax, and the J&J, which in particular is much anticipated because it offers the option of a single dose. Interestingly, the J&J is also in phase 3 clinical trials in both Brazil and South Africa. So that can give us some data on the ability to tackle those variants. On the periphery of all this discussion, maybe not as much on the periphery in Nigeria, but here in the US is ivermectin. This week, the WHO recently released an updated recommendation and declined to endorse this medication. And just 2 days ago, Merck put out a very strong statement discouraging the use of ivermectin. So we have a lot to talk about. To lead us in discussion, we have 3 strong panelists. I'll first of all introduce Dr. Getty Anyanwoke, who's a fellow co-moderator today. By now, she's a familiar face. Getty has become ubiquitous in COVID-19 discussions. I wonder if there's more than one Getty at this point, because I see her on all flyers 7 days a week, 8 days a week. Should be there. Her authority speaks for itself. Her availability is equally impressive. Getty, as many of you know, is a fellow of the Infectious Disease Society of America, chapter chair of AMPA Louisiana, infectious disease committee chair for AMPA, and the medical director for the infectious disease specialist of Baton Rouge, a multidisciplinary specialty clinic also of Baton Rouge. So Getty, welcome. Over to you.
Hello, good afternoon. Thank you all for being here. Without further ado, I'm going to launch in to introduce our first panelist of today. He is Dr. Adefuye. Dr. Adefuye is an infectious disease physician and epidemiologist. He's currently the executive medical director at Takeda Pharmaceuticals. He serves on the Pharmaceutical Industry Consortium on COVID-19 and National Institute of Allergy and Infectious Disease COVID Immunoglobulin Alliance. He's the immediate past chairman of the AMPA Infectious Disease Committee. Dr. Adedeji Adefuye is my boss, and I welcome him to this panel today. Thank you, sir.
Thanks, Getty.
I'm also going to introduce Dr. Obi Nedu, who's a second panelist. Dr. Nedu has been with us from the onset and is a familiar face at this point, as well as the other panelists. He is also a fellow of the Infectious Disease Society of America and also the Center for Tropical Medicine Infectious Disease program director, at Ochsner. He's very involved in COVID-19 clinical trials and can provide tremendous insight into the discussions that we have today. Dr. Anyanwoke will introduce our third panelist, who's also going to give us a brief lecture. But before we get there, we're going to call on the AMPA president, current president, Dr. Chris Okunseri, who's here with us for some time to just make some brief welcome remarks. Thank you.
Thank you, Dr. Oyinke and Dr. Anyanwoke. It's a pleasure to be here. And on behalf of AMPA, the Association of Nigerian Physicians in the Americas, I want to once again welcome everybody, but most especially to give a shout out to the panelists. They have been tenacious, they have been competent, they have provided us with all the knowledge, and we are grateful to them. I'm also going to give a shout out to Getty. Getty, like Aham has just described you, powerful, persistent, available with her time, generous with her time, and the organization recognizes what you are doing, and we are very much appreciative. And like everybody is very much aware, AMPA has content experts, and in these times of crisis or a pandemic, you can see how we have come out in full force to show that within our organization, we have the expertise that is needed for us to be able to help, educate, address concerns and comments. And I strongly advocate that we continue in this spirit of togetherness, of sharing our content or sharing expertise that we all have as a group with fellow physicians here in this country, back again to Nigeria, sharing our knowledge. So once again, I thank you all. And I know that as usual, you're going to have a robust meeting here, a robust conference. And thank you very much for all that you guys do. Appreciate it.
Thank you, President.
Thank you so much. I actually did not know I was not on video the last time. So it's my pleasure to present our presenter today. He is Professor Igor Ofotokun. He's a fellow of the Infectious Disease Society of America. He's done a lot of work with HIV/AIDS. He's a professor of medicine at Emory University School of Medicine. He's member of the FDA Antimicrobial Advisory Committee. He's also a member of IDSA and HIV/MA governing boards. He is a member of the NIH Infectious Disease Clinical Research Consortium Leadership Group, which coordinates all of the COVID-19 vaccines and therapeutic trials. Ladies and gentlemen, I give you our very eminent Professor Igor Ofotokun. Professor, you're welcome, sir.
Thank you, Getty, and thanks to everyone on this forum. This has been a very enlightening forum for me, and all the time that we have participated, I have left the forum just as educated as I came in, really learning from everybody. So today, when Dr. Anyanwoke asked me to do this presentation, I was wondering exactly where to begin, because as Dr. Oyinke mentioned, information about COVID is coming from every direction. We are loaded with information from every angle, in therapeutics, in vaccine, diagnostics, clinical presentation, management. The things are it's just a moving target. And so what I thought I would do today is to raise some points to really guide our discussion. I'm hoping that this will be create an opportunity for all of us to have a dialogue together as we have done in the past, because there is really a huge wealth of expertise, of knowledge in our group. Experts in infectious diseases, experts in critical care, experts in surgery, in psychiatry, clergy, medicine, non-medical expertise that is present here. And we need everything. I mean, COVID touches every aspect of life. And so, so I'm hoping that after this brief discussion, we would have a really great dialogue. Let's see here if I can advance my slide. So COVID, it's like we are flying the plane as we build it. A lot of what we do it's really, things are changing by the minutes. There's really nothing, a lot of all the information, practice guidelines, they change in hours, not even days. So you could go to the WHO guideline today, and then tomorrow what is there is different. The same thing applies to the NIH guidelines. So this plane is something that all of us are just flying it, and we're building it at the same time. New drugs are coming into market, old drugs are falling away, new vaccines are coming, and just things are moving at such a rapid speed. And that is why I'm so excited today that we have been able to put together this forum that brings all of our expertise together and to help really couple this plane for our community while at the same time flying it. So these are some of the outlines that I wanted to kind of use to guide our conversation today. Give a little bit of update about where we are with the vaccines, mention or discuss briefly some of the new findings, updates on the variants, some of the therapeutics, and then miscellaneous issues related to some of the new drugs that or repurposed drugs that are out there. What are some of the data that are out there in support of their use or their non-use? So beginning with the vaccines, these are, as we had mentioned before on this forum, these are the various vaccine platforms. We have about 5 vaccine platforms in general, and these are the nucleic acid platform, the viral vector platform, the viral particle, the protein subunit, inactivated protein. Up until now, there had not been a single vaccine that was approved that used the nucleic acid or the messenger RNA platform. So now we know that the Pfizer and the Moderna vaccine are both RNA vaccine. We talked about the Janssen, the AstraZeneca, and the Janssen or Johnson & Johnson vaccine are both viral vector, and the Novavax vaccine that is now still in clinical trial, it's the protein subunit vaccine. Before now, some of the most common platforms were the inactivated virus vaccine, as well as the viral particle vaccine. So let's see where we are with each of these vaccines. So on this slide, we'll spend a little bit of time orienting you to this, but it provides really a brief summary of some of the nice presentation that OB gave to us when we started this forum. Just a brief summary of all the vaccines that are currently either in use or in clinical trials in the US. So we have the Pfizer and the Moderna vaccine that have been approved. They, well, let's say have the emergency use authorization by the US FDA. These are the ones that are currently being administered in the country. We have the AstraZeneca and the Johnson & Johnson, which have completed their clinical trials in the US. The AstraZeneca just filed their emergency use authorization to the FDA a couple of days ago. And then we have the Novavax vaccine, which I had said earlier on is a recombinant protein adjuvant vaccine. So what is the dosing? Let's take the Pfizer vaccine, start with the Pfizer vaccine. So it's a messenger RNA. It is given twice. There are 2 dosing schedules given 21 days apart. So the storage is requires a cold chain, usually minus 70 degrees, which is not readily available even in many clinics across the United States. It's going to be a difficult one to really administer in resource limited setting. So the clinical trials that led to the emergency authorization, emergency use authorization included about 43,000 people. So the efficacy data is quite impressive. All of you have seen them on the news, 95% protective against infection. 90% protective against severe disease, not a single death in anyone that was vaccinated with this vaccine. The issue of variant, we will elaborate more. We don't really have a whole lot of data in terms of clinical studies looking at whether the Pfizer vaccine is going to be effective against the various variants that are out there. What we know is that when you test them again in the laboratory, it seems like some of the tests does indicate that it does neutralizes the spike protein of the mutant COVID-19 virus, particularly the UK one. Some studies show that there is slightly decreased neutralization. So the Moderna is the one that was developed here in the US from start to beginning by a small biotech company in Massachusetts. It's a messenger RNA vaccine. It is given in 2 doses, 28 days apart. It also required a cold chain, has to be stored in -20. There were 28,000 people that participated in the clinical trial that led to the emergency use authorization. The number of cases of COVID vaccine cases in that trial was 236. Similar to the Pfizer vaccine, 94% in protecting infection, 100% efficacy in protecting severe disease. Not a single person that was vaccinated died from the infection. We now know from just lab assay that it neutralizes the UK, which is the B.1.17 variant that is found in the UK. There's a but with the variant from the South African variant, the B.1.351, there's a 6-fold reduction in neutralization in the lab. Whether this translates to decreased efficacy in clinical setting is yet to be determined. So the AstraZeneca is the Chad adeno-vector virus. In the clinical trials that were done in the US, they were given in 2 doses, 28 days apart. The trial that was done in the UK and Latin America, it was 2 doses given either 28 to 4 to 12 weeks apart. The good thing about this is that it requires regular refrigeration, 2 to 8 degrees Celsius. 11,000 people in 3 studies. There was 131 cases. What we found in this trial was that the efficacy against efficacy in protecting against COVID vaccine infection was 70.4, but 100% protective against severe disease. Not a single person that was vaccinated died. So we don't have any data yet on whether this vaccine is going to be just as effective against many of the variants that we see out there. So the Johnson & Johnson, the Johnson & Johnson or the Janssen vaccine is another adenovirus vector vaccine, AD26 vector. The good thing about this vaccine is that it's a single dose. It does not require cold chain. You can store it in regular refrigeration, but it can be preserved for up to 2 years if it is stored in -20. So the study, which was recently concluded, enrolled 43,000, almost 44,000 people. This was at the peak of the 3rd wave of the epidemic in the US. There were about 468 cases. So the efficacy, remember this is a 1-dose vaccine, was 66% effective in protecting against infection, 85% reduction in severe diseases, no 0 death. And one of the things that we were able to find about from this study, Johnson & Johnson study, is that we know from clinical study, they actually did some studies in the UK as well as in South Africa, there was a clinical decreased efficacy to the variant. So 50%, 57% effective against the South African variant. So which could be an issue with the use of this vaccine. So the last vaccine, which is the Novavax vaccine, this one is still in clinical trial. As of last Thursday, they had enrolled about 20, they had enrolled close to 24,000 people have been enrolled. The plan is to get the study completed by this week, this coming week, either anytime between Monday and Friday, it should be completed. We are enrolling at a rate of about 800 to 1,000 people a day. So it's given 2 doses, 21 days apart, require regular refrigeration. So study that was done in UK and in South Africa showed that it's about 90% or 89% effective again in UK and 49.4% effective in South Africa, 100% protective against severe disease. The efficacy against the UK strain is slightly decreased, and the efficacy against the South African strain is really markedly reduced. So this is kind of really a summary of all of the vaccine. But the take-home message, what I really want us to leave with, the bottom line for all of the vaccine is that they are extremely very effective. So when we started this issue of vaccine development for COVID disease, we were hoping that we would have a vaccine that is somewhere around 60 to 65% effective. So it was just remarkable that the efficacy of the vaccine was 95% for the first 2 mRNA vaccine. So when you see a number 60% effective, 70% effective, these are good in the vaccine world. These are extremely high efficacy number. So, but the bottom line is that they all protect against a severe disease. So if you put all the trials together, people that were given the vaccine, so remember it's a randomized trial. Some people got vaccine, some people got placebo. If you take all the people in all the trials so far that have been given vaccine, a total of about 75,000 people that have received the vaccine product, whether it's the Moderna vaccine, the Pfizer vaccine, the AstraZeneca, Johnson & Johnson or so, there has not been any hospitalization for severe COVID-19 disease. No death in anyone that has been vaccinated. So which is kind of really the key thing, that the vaccine is able to protect against infection, but more importantly, against severe COVID-19 disease, as well against COVID-19 death. And so there's also evidence now, I think this was data that was generated by AstraZeneca, that the vaccine may reduce the risk of transmission. So people that are vaccinated, even if they are exposed to COVID-19, they are not as infectious, they don't transmit the disease. So we're still looking at those data and we're trying to tease that out with all of the vaccines. So that brings us to the issue of distribution of the vaccines. So we have 2 vaccines that are currently in use in the US. We have an AstraZeneca vaccine that is also being distributed in the UK. And we have the Russian vaccine, the Sputnik, or the Gamaleya vaccine, which we would talk about briefly, that is being distributed. As of February 3rd, when I was putting this slide together, in the world as a whole, we have vaccinated, we have given about 180 million shots in 67 countries. So these are the top 5 countries represented on this slide. Israel, for example, has vaccinated over 50% of their population. They vaccinated close to 90% of all their senior population, anyone that is 65 years and older. Here in the US, we have vaccinated about 10% of our population. And as of February 3rd, 35 million doses of the vaccine has been administered. And again, as Dr. Onyeke mentioned earlier on, there's been big issue of inequality, disparity, like all other aspects of healthcare in the US, big disparity in the administration of the vaccine. you can see on this figure here, the ratio of white to black that are vaccinated is about 3. So for every 3 white individual that is vaccinated, there's 1 black person that is that have received the vaccine. And this varies from state to state. You can see the distribution here. Pennsylvania is probably the worst, where you have the ratio of white to black vaccination of 3.8%. We don't have complete data, ratio data on everyone that has been vaccinated. We have data in only about 51% of individuals. From those data, we know that 60% of those vaccinated, it's 60.4% are white, 11.5% are Hispanic, about 6% Asian, 5.4% African American. So there's still a lot of work to be done with educating all communities, particularly the minority communities, about vaccine uptake in the US. I just kind of wanted to also highlight the fact that there are other vaccines besides the ones that are available in the US being developed in China, Russia, and India. And I just want to really highlight the Gamaleya vaccine that is being that was introduced or developed in Russia. This is the Sputnik 5. So the Sputnik 5 is an adenovirus, a vector vaccine. One of the things that the Russians and the Chinese did was that they introduced their vaccine to the general public after the phase 2 trial, before the big phase 3 trial. So the green line on this slide represents early approval and use in the population. So as early as last summer, they were already using their vaccines in the general public. But towards the end of the year, the Russian completed their phase 3 trial, which was published in Lancet on the 2nd of February, this current week. I think it was Tuesday of this week. So 40,000 people were enrolled in that study. The efficacy was 92%. And you can see on this figure here, so the red is placebo and the blue is those who were vaccinated. And this is the rate of incident COVID infection. And you can see there's a huge difference. You know, the vaccine is extremely effective. It does not require cold chain, and it's being widely distributed because it's priced relatively low. I know Mexico is using this vaccine. Many parts of the Middle East are using this vaccine. This is something for us to watch out for. This is one of the vaccines that may actually be readily available, that can be deployed to many parts of the world where resources are limited. So what is next in the vaccine world? So we need trials in special populations, and many of these trials are ongoing: pregnant women, children, and immunocompromised individuals. From data that was released by Novavax this week, we know that the vaccine is not as effective. If you look in the South African data that was released by Novavax, the vaccine is not as effective in individuals that are HIV infected. So immunocompromised patients living with HIV, the efficacy of the vaccine in preventing infection was significantly reduced, was about 49% as compared to 60% in the general population in South Africa. Then there's also the other vaccines that are in work is development of booster vaccine against the variants. So we are assuming that variants are going to occur. How do we develop a universal COVID vaccine that will be effective against a variant? So this will be booster. So this will be booster that will be given to individuals that are that have been vaccinated. The other plan, the other effort that is currently ongoing is the development of what is described as the second generation vaccines for COVID. So this is vaccine that will be universal, that would a target epitope that may not be subject to mutation, mutant variant. So there's a company called Gridstone that has this protocol in development, and those trials are supposed to begin sometime soon. And then, of course, we need to raise awareness and promote vaccine acceptance, particularly in minority populations. So I just wanted to say one or two words about the new variants. So like I said earlier on, most of all of this information are in the general public. These days, you hear more, you get more about COVID from just reading the lay press, reading Bloomberg, Politico, CNN, US Today, and a lot of the scientific paper are being released first in preprint before they are sent to peer review publication because it takes some time. So what we know about the variants, which all of you probably have heard in the news, there are 3 of them currently that is floating around. So one thing that I must say is that variant mutation in viruses is not uncommon. So till date, there have been more than 4,000 mutations that have been associated, that have been found with the SARS-CoV-2 virus. So during the process of replication, RNA replication, it's very prone to error. So a lot of the nucleic acid, the building block of RNA is kind of misplaced. The same is true in human when we synthesize protein, but human have, or mammals have the advanced DNA polymerase that do a lot of the repair. So if there's a misplace of a condom, the RNA polymerase comes and repair the damage. Viruses are very simple. They don't have the mechanism for repair. So mutation is common, very common. So what is unique about this mutation, why it is concerning, is that these are mutations that are being found in the subunit 1 of the spike protein. So remember, the spike protein has 2 important functions. 1, the spike protein is needed for transmission of the virus. 2, the spike protein also is the protein that all the vaccine that we're developing today is targeted at the spike protein. So all of these mutants that we're concerned about, these are mutants. So these are shown, these are the different codons that are shown in red. These are occurring in the spike protein. That is why we are concerned about them. So what are the concerns? That it will change the function of the virus such that it could become more, it may have increased affinity for the receptor binding site of the ACE receptor that the virus latches to in order to establish infection. And the second thing is that it may change in such a way that the antibody that our vaccine produce may not be able to bind to this new mutant. So those are the worries. So you have the UK variant, which is B.1.17. We've had, we are not doing a whole lot of surveillance in the US, but we've had about 611 cases in the US. We have the South African variant, which is the B.1.351. There have been 5 cases that have been isolated in the US. And then we have the Brazil variant, which we don't really know a whole lot about, which is a P.1, 2 cases. Most of the cases are represented on this slide. So the bulk of the cases so far have been from Florida and to some extent California. And then they are scattered, especially the UK variants, in different parts of the country. So the concern with this variant is that they are more rapidly transmitted, and evidence have shown, for example, that the cases, the UK variant is about 50 to 70% more transmissible than the original Wuhan variant. The other case is that they may cause more severe disease. We don't know this for sure, but there's one study from the United Kingdom that the UK variant actually led to about 35% increase, 35% increase in fatality with COVID-19. That is just one study. So whether that is true or not, we don't know. And then, of course, the one that all of us are really worried about is that it may lead to escape vaccine protection. The data so far shows that even though people may be infected after they have been vaccinated, it does look that it does protect, the vaccines still protect against severe illnesses and death with this mutant variant. And then the fourth concern is that it may lead to another surge, another wave of the epidemic. So we know from data, the Novavax data that was generated by Novavax in South Africa during their vaccine trial, that 30% of all the people, first of all, 92% of all the infection in that study were from this new variant, the South African new variant, the B.1.351 cases were responsible for 92% of all the infection. But the other thing that was remarkable was that of those who were infected, a third of them already had evidence of infection with the original variant of the virus, the Wuhan variant. So it does appear that even after an initial infection with the original variant, original strain, one could still get a new infection with this variant strain. So which is concerning that it could cause new wave or surge of this epidemic. So I'm just going to switch briefly to the therapeutics. I'm just going to highlight the mainstream therapeutic agents that are currently available. So we can divide the therapeutics into 3 or 4 main categories. So we have the antivirals, we have the immune modulators, we have the monoclonal antibodies, and then we have the others. So there's a whole slew of other agents that are out there that are being speculated to have activities against COVID-19. So let's first of all talk about antivirals. The only antiviral that has received FDA approval in the country, in the US, is remdesivir. So many of you, if you followed the Ebola epidemic in 2012, 2013 in West Africa, remdesivir is a nucleoside analog. It was first developed for the treatment of Ebola. So the trial for Ebola was not successful. Remdesivir was found to be non-effective in treating Ebola. So all of the studies at least funded by the federal government to evaluate remdesivir were the ACT study. So the ACT is Adaptive COVID-19 Therapeutic Trials. So in Act 1, remdesivir was compared against placebo. The main outcome that they were looking for was recovery time. So during the time of Act 1, that was during the period of the first surge of the epidemic. We had people dying in a Lombardy region of Italy. We have New York was locked down, massive death. And so the question was, how do we get a drug that can reduce the recovery time? So it wasn't powered to actually look at mortality. And so the bottom line of remdesivir, when given to hospitalized patients with COVID-19, it led to reduction in recovery time by 5 days. So that is the bottom line. It had very minimum, it had a tendency towards reduce mortality, but it was not statistically significant. So the benefit of remdesivir is that if it's given to people with moderate to severe disease that are hospitalized, remdesivir reduces the recovery time by 5 days, which is significant. So you could be discharged 5 days earlier. And at that time, for those of us who were taking care of COVID patients, that was a lot. The hospitals was filled of infected COVID-19 patients. So if you can reduce their hospital stay by 5 days, people thought that was significant. So the second trial of remdesivir was Act 2, which has been completed. So in Act 2, remdesivir was combined with baricitinib. So baricitinib is an immune modulator. It's a JAK1 and JAK2 inhibitor that is used for treatment of things like lupus and what is this disease, skin disease that causes plaque. So it's used for many of these autoimmune disease.
Psoriasis.
Yes, psoriasis, yes, psoriasis. So remdesivir was combined with baricitinib. So it was remdesivir plus baricitinib plus remdesivir alone. Again, the outcome of that study was, the main outcome was recovery time. So when you add remdesivir to baricitinib, you reduce your recovery time, by 8 days, which again was very, very, very significant. But again, there was no mortality benefit. And then the third study was the recovery study, which was conducted in the UK. It was an open label study that looked at dexamethasone plus standard of care. So over 40,000 people were recruited in this study. So then dexamethasone was given to some group, and then others received just whatever the standard of care was for that hospital. Again, this was moderate to severe cases of COVID-19, and there was in general 20%.
Can you reduce that music, please? I'm on Zoom.
Yeah, in general, there was 20% mortality benefit. So this is the only study where the recovery study with dexamethasone that really showed mortality benefit. So today, many guidelines in terms of therapeutics in most hospitals, and I'm sure many of you are aware of this, give a combination of remdesivir plus dexamethasone. Again, these are for people with moderate to severe disease. Dexamethasone was actually had a detrimental effect in people with mild disease. So if you're sick enough, you're not sick enough to require oxygen, your oxygen saturation is not low, treating with dexamethasone led to significantly higher rate of mortality. The same thing applies to remdesivir had no benefit in people with mild disease. So if you don't require oxygen, if you don't require mechanical ventilation, none of these studies work. So this is kind of what I have said, represented here in the graph for Act 1, Act 2, and the recovery study. So I won't spend too much time on this because these are not controversial. These are like standard of care. And if you ask me, dexamethasone is probably the key drugs in the treatment of COVID-19 because we know it reduces, it improves recovery time. It also has mortality benefit. So the mortality benefit is even bigger, it's higher, in people with more severe disease, people that are admitted to the ICU and in ventilation and being ventilated. So if we go to monoclonal antibodies, this is really where I want to draw your attention to the monoclonal antibodies. Unlike the drugs we've just mentioned earlier on, monoclonal antibody work more in people with mild disease. So the study in monoclonal antibody was done both in outpatient setting and in inpatient setting. There was no benefit when you use monoclonal antibodies in people that are sick in hospital, on oxygen, or in mechanical receiving assisted ventilation. So people with severe disease, monoclonal antibodies don't work. So monoclonal antibodies, they target the virus and they neutralize the virus just like vaccine would do. So there are 2 of them currently approved or have received emergency use authorization in the US. So we have the Lilly product and we have the Regeneron product. So here are the data that led to their approval for emergency use authorization. These were outpatient study, and what they were looking for was recovery, symptom score, and then prevention of hospitalization. And then the third parameter they were looking for is viral clearance. And so what you will see, if I could go back, so if you look at the symptom score in this, if you can see my diagram, the first diagram, I hope you can see my cursor. You see that the red arm here is placebo, and the blue is the product, monoclonal antibody. So from day one of presentation, you start people on monoclonal antibody, you see a marked improvement, fast, rapid improvement, resolution of symptoms in the treated individuals compared to the placebo individual. So rapid improvement of symptoms with the monoclonal antibody. The second thing is, does it prevent hospitalization? So if you take it as outpatient, will it prevent you from being hospitalized? And the answer is yes. So people who received placebo, no monoclonal antibody, rate of hospitalization was 6.3%. And people who were treated with monoclonal antibody, rate of hospitalization was 1.6%. This is almost like three to four-fold reduction in the risk of hospitalization. This is with the Lilly product. And if you look at the Regeneron product, they look at the rate of viral clearance. So what this means is that you're able to quickly clear your virus so you are not infectious to other people. And also the faster you clear your virus, the better you also improve in terms of symptoms. And placebo is in gray, and then different doses of Regeneron monoclonal antibody, the green and the blue. You can see that the clearance of the virus over a period of 7 days was more rapid when people were treated with the monoclonal antibody. So in our experience here at Emory, we have infused hundreds and hundreds of people with either the Lilly product or the Regeneron product. I don't think we've had any one case of somebody who was treated in our experience that have died or that have been hospitalized. And the key is to be treated early. So all my patients, as soon as you have symptoms, get tested, get linked to monoclonal antibody. So the earlier, usually I would say within the first 4 days, the earlier you present for treatment, the better, the more effective the monoclonal antibody are. So they lead to faster recovery, reduced hospitalization, rapid viral clearance. And then there's a study that has not been published that actually shows that they can be effective in prophylaxis. So this was what the study was done with the Regeneron product. It's a household contact. So if you had COVID and everybody in your household is then given monoclonal antibody, and this was given subcutaneously. So all of these other studies were infusion. So this, you go to the people's house and you give them this monoclonal antibody subcutaneously, 80% protection against infection. So it was really good as a preventive measure against people who have had contact or have been exposed to COVID-19. So I will be remiss not to mention these other agents. There are several slews of other agents that are out there that is speculated to have activities against COVID-19. For the purpose of the discussion today, I just limited myself to 2 of them, colchicine and then ivermectin, which we have discussed extensively. So the problem with all of these studies, ivermectin, colchicine, the antihistamines, antipsychotic drugs, is that a lot of the studies have been either small, non-randomized, and the endpoints have been all over the place. But recently, I think 2 weeks ago, this study of colchicine is used as an anti-inflammatory drug that is used for the treatment of gout, looked at over 4,000 participants in Canada. So this was an observational study. And so what they observed in this study of colchicine that was given to people with COVID-19. So remember, colchicine, the value of all of these drugs is that they are oral drugs. So you can give them in the outpatient setting. So it led to 25% fewer hospitalizations, 50% less need for mechanical ventilation if you were hospitalized, and 40% fewer deaths. So when you look at this data, it looks very, very, looks good. It looks compelling. And as a matter of fact, in Canada, they are actually moving ahead to include colchicine as part of their treatment, in their treatment algorithm for COVID-19. But again, this is a non-randomized trial. And then the case of ivermectin is being, a lot of study on ivermectin. So I included here a meta-analysis that was put together by Andrew Hill. Andrew Hill is a clinical pharmacology in Liverpool, who I've known over the years through our work in clinical antiviral research. So what he did was to look at all of the studies, or several of the studies, both observational studies, randomized trials that have included ivermectin. And so what he was, when he pulled them together in this meta-analysis, he was able to show that you see 87% mortality benefit with ivermectin. And then he was also able to show that we have 43% higher rate of recovery. And in another slide, which I did not include here, was that there is faster clearance of a virus. Again, some of these studies are so small. Many of them, the endpoints are just different. The doses of ivermectin that were used are just different. And we know that COVID-19 disease is a disease that people can recover. If you don't randomize people, you never know. People can spontaneously, overwhelming majority of people with COVID-19 spontaneously recover. So different people are taking this data differently. Some people are saying ivermectin is effective. They want to use it. Others are saying we need larger studies. Others are saying the effect size are so small in some studies. These studies are so non-uniform, you cannot really compare them in meta-analysis. So the jury is still not in on the case of ivermectin. I think Hiran mentioned Merck put out a statement. Merck is the manufacturer of ivermectin, disclaiming their support for the use of ivermectin for the treatment of COVID-19. They don't think there's any data. The NIH guideline committee panel is reviewing ivermectin data again. Last month or a few weeks ago when we met, they were still strongly not recommending it. I think what I hear is that they are neither in favor or for or against ivermectin at this time. They are seeking more data. So this is one of my last slides, the issue of the burden of COVID-19 in Africa. So as of this week, we've had roughly about 3 million of the 100 million cases of COVID-19 in the world. I mean, compared to the rest of the world, this is a relatively small proportion of the disease. We had thought when this epidemic broke about this time last year that Africa was going to be rampaged by COVID-19. So there's been a lot of speculation as to why this is the case. There are speculations that this may be related to malaria infection or a use of anti-malaria drug, BCG vaccination, the weather, and maybe this is not true data, we're not testing enough. So we don't know where the truth is, and I'm sure this is going to be something that is widely investigated. But there was this study that was published a few weeks ago. Again, this was published in the BMJ preprint, and this was done in Lusaka. So what these people did was to examine all of their, did a systematic postmortem surveillance of deaths in the Lusaka area. So they look at death both from the major city and also death from the rural communities around. And so what they did was, these were people who died not knowing their COVID-19 status. They were not tested for COVID-19. So in this autopsy, they swabbed them and they did PCR testing for COVID-19. What they found was that 20% of the couples who died for other reasons other than COVID-19, 20% of them were COVID-19 positive. So what this study suggests, if it's confirmed, that in many parts of Africa, and I think that is true also for here in the US, because I don't think everybody, even though we're testing a whole lot, not everybody is being tested, that the prevalence, the rate of this infection is being underreported in many parts of the world, including Africa. So I'm going to stop here, and I think I've given us enough to think about and to really generate a robust discussion around. So I'm looking forward to this discussion and the contribution of the other members of the panelists and the entire group. Again, thank you for your attention, and I really appreciate the opportunity to really dialogue with us in this forum. I'll turn it over to the moderators.
Thank you so much, Dr. Fotokun. As usual, you bring it, the knowledge, the presentation, illustrations on point. Excellent lecturer, as we've seen. So are the rest of our previous lecturers. So thanks once again. Everybody is welcome to participate in this discussion, which will be led by our panelist, Dr. Ofodokun, we just heard from, and we have two other panelists, Dr. Nedu and Dr. Adedeji. Myself and Gertie Anyanwoke are the moderators, and we would selectively, using our best discretion, take questions from the chats and bring them to the main forum. With that in mind, it's probably best if we organize it based on the order in which Dr. Fotokun presented. So we can start off with some questions pertaining to the vaccines, then move on to his brief discussion on distribution and the inequities, and then look at some things on the variants, and then therapeutics, which is what he closed out with, and of course, any other miscellaneous things that the audience wants to hear about. So once again, we'll encourage everybody to use the chat, and myself and Getty will continue to monitor it and pull questions from there. You could also raise your hand if you want to speak, and again, we would use our discretion. So Dr. Fotokun, one question that keeps coming up is, what exactly does efficacy mean? Are you able to elaborate on that? You know, we hear 95%, and obviously it sounds impressive, but can you just elaborate a little bit on how that is calculated? I know you had some previous slides on that. If you have it, you may pull it if it's easily.
Yeah, I can. Let me do this.
Okay, and while you pull that, we'll come back to that. Perhaps Dr. Anyanwoke can make some comments on the pregnancy issue. That is something that continues to come up and is one of the reasons for vaccine hesitancy.
Yes, yes, I'm going to make some comments about that. There's I think, is it Miss Susan Baker Small? She asked the question about S protein and fertility in women. So now I understand why people are worried about the COVID-19 vaccine and talking about pregnant women. There is a difference between protein S and S protein. See, all this time I've been telling people, like, where did they get this information from? Now I finally get it. Protein S and S protein are two different things. When we talk about S protein on COVID, we are talking about the spike protein. When you look at COVID and it's round, you see those spikes that come out? That's the spike that the COVID virus itself uses to get into the cell by going through our ACE2 receptors. So we call it S protein for short instead of spike protein. The protein S that can become an issue for fertility is in our coagulation system that functions. That is a different issue, and it has nothing to do with the spike protein, which we nicknamed S protein. So
Ms. Baker, I hope that you have your understanding of the differences between protein S that could potentially cause fertility issues and S or spike protein. They're two different things. I hope that answered your question. Thank you.
Yes. So Getty, if I may add to that, I'm glad that is a very clear explanation. I'm glad you brought that up. The other thing to remember is that COVID-19 does two things to your body. It causes inflammation. It also leads to coagulation disorder. So protein S is one of those coagulation protein in the body. So COVID-19 really, the infection itself disrupts, your coagulation system. If anything, it's not that there's any evidence that post-COVID-19 effect is infertility, but if anything, the virus, due to the way the mechanism of disease, is more likely to affect your protein S and not the vaccine. So this speculation that the vaccine would affect fertility is baseless. It has no basis in science at all. There's nothing. And so remember the trials that we have now given close to 100,000 people COVID vaccine in the world. Many women have become pregnant after they received COVID vaccine in clinical trials. And we are now giving it to people in real life. We have vaccinated over 108 million people. Overwhelming majority of the people that have been vaccinated are women. There is no scientific basis. There is no epidemiological evidence that COVID-19 affects fertility in women or in men.
Okay, thank you. So that point there that some women got pregnant after receiving the vaccine in clinical trials should completely refute that concern. So Dr. Igor, you have your slide shared. Are you in position to guide us on how we understand efficacy?
So efficacy of a drug, when we conduct a clinical trials, we always have endpoint. And so those endpoints are very defined. And so those, in the case of COVID-19, the endpoint for most of these clinical trials, for the vaccine trial, is prevention of new infection. So those endpoints are the ability, the rate, the proportion, to the ability for the test product to achieve those endpoints in clinical trial is defined as the efficacy of the drug. So the efficacy is different from the effectiveness. So remember, in clinical trial, it's very well regulated. There's a protocol. You are instructed to behave in certain ways. You cannot take certain drugs. You cannot do certain things. So there are really guidelines. There are instruction. When you are in a clinical study, you have to follow the protocol. But in real life situation, the ability of the drug to do what it's meant to do in real life situation where there are no guidelines, you're not regulated. People may take one dose, and then the second dose they may not take. They may take one dose, and the second dose they may take more than 21 days or maybe 12 weeks later. So the situation in real life, like now that the vaccine is being deployed, is different from the regimented or controlled environment of clinical trials. So the ability of the drug to achieve the endpoint, the desired result in the real life situation, that is where people are not randomized, is the effectiveness of that drug. Sometimes efficacy and effectiveness may differ because the environment under which the drug is being administered in real life situation is different from what happens in clinical trial situation. So the goal is that we want both efficacy and effectiveness to be as close together as possible. So the efficacy gives us a gauge of what we think the drug will do in real life situation.
Okay, thank you. We can bring in Dr. Adedeji on this next question, and it has to do with the viability of the vaccines. How can we be confident that the vaccines can give long-term immunity, considering that we've only had a short duration of trials? And secondly, with the J&J saying that their vaccine can last in storage for up to 2 years, can you explain how they can predict that, given that they obviously haven't had the vaccines that long?
Okay, thank you. I think this is actually a question that's come up several times in terms of, how can what do we know about the durability of the, of the response, or the durability of the protection that you get, after you have had the vaccines. The bottom line here is that, as we do not know how long the protection that you get from natural immunity, which is essentially is when you're infected and you recover. We don't know now, and you probably have heard from both Igor and Obi, from previous sessions, that the clinical trials have not stopped. We are still continuing to follow the individuals who participated in those clinical trials. And, we're still doing, regular, antibody testing, and they're all going to be followed for a period of 2 years to actually see whether the level of antibodies is still actually sustained up to that level, because you have to have a certain level for it to be protective against, getting infected and actually having severe disease. So we're going to follow, for 2 years and actually see where we are at the end of the 2 years. So the answer to that question right now is we don't know, which is the reason why we're still following these patients, and we're going to follow them for the period of 2 years after they actually completed all the necessary measures in the studies. So that's one. The second question about you can actually have the vaccine, still be able to still have it viable for a period of time, is that different manufacturing processes are used for, all drugs, not just vaccines. So the manufacturing process is actually what determines the level or the temperature, the environmental condition in which you actually will put the vaccines. So, and there's also one thing that is actually done. Every pharmaceutical company actually does that. When you manufacture products, vaccines included, you actually keep a small proportion that are supposed to be a proportion that you actually will keep and follow over time to be able to measure stability, to be able to, that is how that's how, apart from your PK that you do, which is pharmacokinetics that you do during development, which gives you an idea of metabolism, which gives you an idea of the half-life, of all these different products. You also do that to actually see, and that's what actually eventually gives you an idea into how long the vaccine can last, because they are exposed to different temperatures, excursions, and everything to be able to make that determination. It is by doing that that you might be able to make a determination, okay, this can actually last for so long if it's refrigerated. If you go above a certain temperature, because of the nature, these are biologics, and this actually can get denatured, and then you will not get the benefit that you're supposed to get from them.
Thank you. So we'll bring in Dr. Nedu for the next question, which is, do the vaccines effectively prevent transmission? And do the vaccines prevent, how effective are they against asymptomatic infection? So there's two questions there.
Yeah, thanks. Those are great questions. In terms of transmission, we still don't have enough information about that. I think Dr. Ofotokun presented some preliminary data with one of the vaccine candidates that it possibly may reduce transmission. But we don't have enough information yet. And so the guidance still is, even after you're vaccinated, it's still important to, oops, sorry. I just realized I'm muted, I'm sorry. So what I was alluding to is the fact that we don't have enough information on transmission potential for those who've been vaccinated. Dr. Ofotokun presented some preliminary data that one of the vaccine candidates may decrease the likelihood of transmission. But at the moment, we still don't quite fully know whether those who are vaccinated are able to transmit or not. And so from that standpoint, even after one is vaccinated, it's still important that one wear a mask and that one continue to socially distance. And I suspect that will still be the case until such a time when community spread of COVID-19 has dropped below certain levels. Then the next question was in reference to asymptomatic infection. What most of the trials have focused on was really sort of symptomatic. People get symptoms and then you test them for COVID. And in some ways, that's probably a little bit more of a benchmark in terms of protection from clinical disease. And then they also looked at protection from severe disease and hospitalization, and then protection from mortality. And certainly the vaccines all showed tremendous benefit from that standpoint. And so and I just wanted to kind of just comment about the variants, just to add a little bit to what Dr. Ofotokun alluded to. We're going to get more information from the variants, but I don't want people to kind of lose hope as far as the vaccines are concerned. As was mentioned in that great presentation, mutations are known to happen with viruses. Sometimes they can affect vaccine efficacy. I think what one would look for long term would be, whether or not there's still some reduction in clinical disease and hospitalization and death, even with the variants, and then whether or not we may need to make some modifications to the vaccinations in the future. But I think it's still extremely exciting that we have vaccines available. This pandemic has killed so many people, as we all know. And so we certainly need all the tools that we can to try to bring this to an end. Thanks.
Yeah, and if I can actually add something quickly to that, if one, even for the variants, we still see that the level of efficacy is probably still, below. It's still well, above 55, 57. I think that's the least that I've actually seen. When you put that into context into the flu vaccine that we all take all the time, the current flu vaccine that we have, later part of 2020 to 2021, is only 45% effective. Okay, when you put it into context, we're not complaining about the flu vaccine just being about 45, and we're all still rushing to take it and everything. And even with the variants, I don't think any of the vaccines actually have levels that is as low as that of the flu vaccine. So just putting it into context, so we still have an understanding of where we are with this.
Okay, thank you. So that's a very important point Dr. Deji just brought to the table, because it seems somehow that the medical community might be, to a large extent, underselling the current vaccines, considering the impact of the epidemic or pandemic, I should say, and the rapid ability to develop something with 95% efficacy. The messaging probably could be more positive, more encouraging, but perhaps when we talk about still wearing face masks and still social distancing and still keeping schools closed, then it allows the opportunity to question why bother, why even give the vaccine in the first place. With that in mind, perhaps Dr. Ofotoku, the two questions. One is, how familiar are you with the J&J study that showed that did PCR testing as part of their clinical trials and showed that after about 3 weeks, up to 65% of patients were protected based on PCR testing, which then implies that a single dose could have significant efficacy. And it's believed that the longer the person is from the administration of the vaccine, the stronger the immunity builds. Are you able to comment on that, Dr. Igor?
So the question is,
so we've been talking about transmission up to this point based on clinical symptoms, but this other study was of a PCR test, which showed up to 65% prevention from people who got the vaccine after only 3 weeks.
Yes, I get them. I think it was the J&J. I don't know if it was the J. I think it was the J&J that alluded to the fact from those data that their vaccine may actually protect against transmission of the infection. So I think that was the first time we're beginning to see that. So there are so many things we're looking for in terms of the benefit of the vaccine. Of course, we want to prevent infection. 2, we want to prevent hospitalization. We want to prevent death. But the other benefit is that we want to break the transmission cycle. We want to reduce the ability of people to transmit the virus. And so I think it was the J&J study that came first with this data suggesting that there might be this added benefit that if you are vaccinated, the risk of transmitting the infection, even if you are infected, may be significantly lower than if you were not vaccinated, which is, if it's confirmed, and I think a lot of the other studies now are beginning to add this component to their study. So if this is confirmed, it will be a welcome news, because all we want to really do is to break the cycle of transmission. I don't know if I addressed your question.
You did. Actually, I teased it out even better than the question was asked, which is that if, and just to recap, if it prevents PCR positive testing, then it implies that it can prevent transmission, not just from monitoring who has symptoms, but we now know that you can't get a positive test, which means you can't transmit it in the first place. The other vaccines looked at symptomatic patients, meaning that some of them may be carriers. So thanks.
The bottom line, I mean, all of this information can be confusing even to those of us who are infectious disease experts. My simple mind, the way I look at it, is that we want to prevent hospitalization, we want to prevent death. All the vaccines, regardless of what the efficacy number is, even in population where the efficacy number, like in South Africa, where the efficacy number was low, 49%, in some part of the population, nobody died, nobody was hospitalized. I think that should be the key message.
With that in mind, I'll throw this one to Dr. Nedu. If for all the vaccines so far seem to show good limitations in disease spread and symptoms severity with just one dose, the United Kingdom has gone to a public policy of just one dose for now, with the second dose being deferred. What are your thoughts in terms of trying to get as many people vaccinated? Is that a reasonable option to consider, to vaccinate as many people with just the first dose and then come back later and do a second dose? And then also looking at Nigeria, where the logistics of 2 doses might be an issue.
Yeah, so my first part of the answer will be to just state what the CDC says in the US in terms of the vaccines we have available here in the US are required 2 doses. Right now, the CDC still says you should get both doses. That's because we have, more data related to both doses. What the CDC does allow for, though, is a delay in the second dose because of the distribution and so forth. So after the first dose, there can be a delay of up to 6 weeks in terms of getting your next dose. And so that is sort of allowed and accommodated. And in general, I forget who mentioned it at a previous session, but the way the immune system works, the way the immune memory works, after you get your first dose, the memory is there. And so if there's a delay, just in terms of how the immune system works, it probably will still be effective. As you alluded to, the only vaccine that has really been kind of systematically studied in terms of just giving one dose is the Janssen or the Johnson & Johnson. That seems to show some good efficacy, preventing death and preventing hospitalization, as was very nicely presented in the presentation earlier. That doesn't require as much of a cold chain, so to speak, and so transportation and storage seems fairly straightforward. I'm sort of more someone that would let the data guide me. I think if I were to make a recommendation for a resource-poor place where there may be constraints on getting second doses in, then I might choose something like the Johnson & Johnson one. To date in the US, we just have the mRNAs that have been given conditional authorization from FDA. And then Johnson & Johnson, I understand, has applied for that. So that may become available here in the US. Certainly, that's a very promising option overseas. The Novavax also is a traditional protein subunit vaccine. Doesn't require, requires the same storage and transportation as vaccines that are already in use in Nigeria and many parts of Africa. So that may end up being a good option as well. So I would favor kind of going with the data rather than speculating, particularly as at a time when we're still collecting information. And I just kind of want to make a clarification. These clinical trials have not ended. So some of them have stopped recruiting people, but the trials are still ongoing. And so these people will be followed for up to 2 years. And so recruitment has stopped for some of the vaccine candidates. For some, but the trials will still be ongoing for 2 years, and we'll get more information. And hopefully, as we get more info, we'll be able to answer some of these questions.
Okay, so Dr. Nedu says follow the data and let it guide us. And the deviation from that that was found in the clinical trials in the United Kingdom could be additional data that we can then use to see whether it makes sense to embrace that. Also, Israel is aggressively gathering data and injecting its entire population. So we'll have some great data from there as well. We'll take one more question from the chat pertaining to the vaccine specific questions. And then Damien has his hand up. So the question from the chat was, is there any evidence of rheumatoid arthritis being a consequence of these vaccinations? I personally haven't seen that, but someone on the chat seems to have seen it somewhere. Perhaps Dr. Deji can chime in there.
One thing I will say is that one of the things you get with the COVID illness itself is an exaggerated immune response, which is actually, of course, you see the cytokine storm, you see all these other things that actually develop as a result of that exaggerated, immune response, which is one of the reasons why dexamethasone, happens to actually curtail, some of those responses. I personally have not seen this, but what I will, I'm going to ask both Igor and Igor to respond. But what I do believe is the fact that in the process of the immune response, when you actually understand that some of these are immune-mediated or immune-modulated diseases, rheumatoid arthritis, psoriasis, and the rest of them, I will not be surprised, if as a result, somebody who has COVID actually has this actually, been, maybe it's a resistant, illness or disease that actually been with that person. And maybe as a result of the immune response, it actually pushes it out. I do not believe that it's the COVID vaccine that is actually responsible for this. Contrary to that, I will actually think it's the exaggerated immune response related to COVID disease that's actually responsible for that rather than the vaccine. So, I want to hear what both Igor and Obi probably will say.
Okay, Dr. Igor, any comments on that?
Yes, I commented on the chat that we now we have had 75,000 people that were given the various vaccine product, and we've watched them very closely. So autoimmune disease is not one of the known side effects or reported side effects of the vaccine. Again, and now we are still learning more about the vaccine. Now we have the phase 4 post-marketing surveillance, and over 100 million people have received it, and it's not being an issue. On the other hand, as Deji said, we are also beginning to study, understand what they call the post-acute COVID syndrome, or the COVID long haulers, that in some cases, people are reporting unmasking of autoimmune disease, especially lupus, with following attack of COVID-19 disease. Whether that is true or not, whether they would have had those before with or without COVID, we don't know. But the vaccine definitely is not associated with autoimmune disease that we know of.
Thanks for the clarity there. Damian Ejigiri has had his hand up for a while, so we'll take his question, and then there's one more question on vaccines from Amadou Diallo. So Damian, you go first.
Thank you very much. I want to express our sincere thanks from the community to the panelists. My question is, because of the limitation of the various vaccines, do you recommend after 1 year that those that have received the first or the 2 vaccines should go back, or wait for a boost system? Thank you.
Okay. Getty, do you want to comment?
Thank you for the question, Professor Jigiri. I don't think that we know that right now, because like COVID is new, it's novel, vaccination to the general community just started. We have to have enough data to make those decisions. I know that there are immunologists that have looked at how long they think that the neutralizing antibodies can last. There was a study that was published, and I think even New York Times carried it. It was said to be between 6 to 8 months. We don't know for sure. That's the truth. We'll have to wait. I know that the vaccine, the trials that are going on with Pfizer and Moderna, they still have like a phase 4 going on, and that will run for, I think, at least 2 years. They'll be able to tell us more, but we will just have to wait and see. Also, because of these new variants that are coming, they're talking about doing bivalent vaccines or polyvalent vaccines. Everything is like a moving target. We're just watching and following it, just the way the whole community is watching and following it. I hope that explains it, Professor Ejigiri.
Thank you very much. It does.
Thank you.
Yes, there is also, let me just add to that, something that Obi actually was talking about previously, about the vaccines. And, I'm happy that you actually talked about this, possibility of bivalent vaccines. We will know that somewhere down the road that even for the J&J vaccine, there is actually a study that is actually just started to see whether response can actually be enhanced by giving a second dose, even though now the recommendation is that first dose. So that just tells you that, things are changing. And as Igor presented, information today might become obsolete tomorrow. So, things are changing, on a daily basis. And the only thing everyone can do is to continue, to be on top of new information as they come out.
Thank you. So as Dr. Ego said, we're building the plane as we fly it. It's very apt description. So Amadou Diallo, if you can hear us, you can go ahead and unmute your mic. Tell us where you're calling from. It's not a Nigerian name, so we're curious. You're welcome, and then you can ask your question. Amadou Diallo. Okay, if you are ready later, you're welcome to ask the question. I'll go ahead.
I'm having trouble with my sound. Can you hear me?
Yes.
Yes, I have a question. I'm from Senegal, and the government has decided to go with the Sinopharm solution. So, do you guys have any feedback on that specific product?
Okay, thanks. Any of our panelists is welcome to comment on that.
Professor Ego, do you have any contribution about the Beijing, I think it's from China, Sinopharm vaccine?
Correct.
So what I know is that it's not different from what we know about the other vaccines. And I think the efficacy data that was reported, it's slightly lower than what we have seen. I mean, we are used to 90%, 94%, 95%. I don't remember the exact number, but
it was 79%. It was 79%.
79%. So, but I don't look at those numbers because they are not, the key thing is that all of them are, they are a spike protein. So they lead to antibody production against the spike protein. And we know that is a very effective target. So any vaccine that will lead to antibody production against the spike protein, I expect them to be effective in protecting against severe disease and in protecting against death and hospitalization. And that seems to be, all the trials that are coming out, that seems to be the kind of the uniform theme. And so I think it's, I mean, the Chinese, they had enough confidence and trust in it that they are vaccinating their population with that vaccine. So I have no doubt that it would work just as well.
Yeah, the only thing that, I will add is that you see the platform, Professor Fotokun showed at the beginning all the different vaccine platforms. This is just one of very few vaccines that actually uses, that is actually inactivated. Mm-hmm. So it's different from all the other ones. I think the vaccine is called Sinovac. And, it's actually, it's an inactivated form, which essentially means inactivated means it's not a subunit. It's not the mRNA or DNA of the virus. Is a complete virus that actually has been killed to the extent that it will not be able to provoke an infection, but it still contains the genetic properties that the body will recognize to be able to produce the relevant immunity so that it will be able to fight, fight the infection if it comes. And just like you heard, the efficacy is actually, in between what you see with, the AstraZeneca, and, it's around, I think it's around 79, 80, and, and just like Dr. Fotokun said, if the Chinese have enough confidence in it to vaccinate their whole population, you want to believe that it's actually, it's actually effective. The other thing to note, and which is something that probably will be important on the long run for African countries, is that the Chinese and the Gamaleya Sputnik 5 actually happens to be the cheapest of all the vaccines that are being rolled out.
Right, and unfortunately, that seems to be a very heavy deciding factor why the government decided to go that route. I just hope that there would be some form of bridge in terms of data sharing with the African community at large, so that very much like we are doing on this forum, sharing ideas, that the clinical data would be shared as well, so everyone can benefit from it.
And Amadou, while you're there, if you can just give us a little bit more insight into how far Senegal has gone. Are they still in the procurement process, or are they already inoculating people?
The answer would be both. They are starting their own in-house bootstrapped clinical trial, and are in the process of acquiring enough dosage to, I think, vaccinate about 10% of the population. And the level of transparency lacks, and that's another discussion. But that's where we are at this junction.
Okay, well, thanks for the useful information.
Okay, I have a question for Dr. Nedu. Ms. Obiagele Naji says, I completed my Pfizer vaccine after 2 weeks of my second dose. I did an antibody screen, which came back positive. So is the antibody test a quantitative or qualitative test? How is antibody level measured, and does the level determine how long one is protected?
Good. That's a very good question. The first thing I just want to say is that, commercially available antibody tests don't all necessarily test for antibodies to the spike protein. And so one thing that's come up is people say, I got the vaccine, should I go get antibody testing? The answer is generally no, because, some commercially available tests test for other protein components of the COVID vaccine. And then in terms of the question of qualitative versus quantitative, so generally each commercial lab will have a set cutoff, and then a titer above that cutoff, they will define that as positive. And that's as much as I can say. And so that's probably what the lab that you went to did. And then they essentially named it as positive. But kudos on getting the vaccine. Yeah, sorry.
She wanted to know if the antibody level measured, and does that determine how long she's protected?
So I would say no, not necessarily. We don't know how long one is protected just yet. We don't know how long those antibodies persist. We don't know how long the antibody component and the cell component of the immune system last in terms of the vaccine. We know that in the short term, it provides protection. We think, or at least I think, that it will provide some protection even longer term, but we don't know exactly how long that will be. And that's a very good question.
Okay, the next question.
No, let me quickly add something to that. We need to understand that the protection is not just about the neutralizing antibodies. We have the team, the TMMM, memory cells and everything that actually remembers the vaccine and the virus when you make contact with the virus next. So it's not just about the antibody, it's also about the T cells and everything. So that is, the protection is not just about the level of the antibody. So I think that needs to be made clear. So you're not basing everything on just the level of the antibody.
Okay, so the next question is, what's the opinion of the panel on the effectiveness of the Oxford AstraZeneca vaccine in over 65-year-olds? Different regulatory bodies have contrasting opinions. So, Prof.
My sense is that I think what I was hoping to achieve in this presentation is to draw people away from looking at the efficacy data that is being thrown around, 95%, 94%, 92%, 66%, 79%. So that is the efficacy against prevention of new infection. What we're really interested in is severe disease and death, severe disease that leads to hospitalization. If you use that as your benchmark, if that is your metrics, all the vaccines are performing very well.
Exactly.
Yeah. Regardless of the age, I mean, if you look at all of the vaccine, if you look at age older than 65 or less, men, women, black, white, if you look at across, all of them are able to prevent severe disease and death. And I think that is probably the ultimate goal. If you get COVID and you don't have the sequelae of COVID, that is really the ultimate goal. And that is my benchmark for all of the vaccines. Yeah.
Dr. Ijoma Nwankwo has a question. May I suggest that the statement, this is a suggestion, that the statement on one-dose vaccine policy in the UK be verified. I'm not sure that I understand that, but it's open to our panelists. Any takers? I think what she's saying is, you know how we have 2 doses, and in the UK, they decided to at least vaccinate people with the first dose and delay the second dose. Is Ijoma Nwankwo here so that she could explain further?
Okay, I can make a quick comment on that. So I happened to read up on that and found it very fascinating. Essentially, the UK equivalent of the CDC saw the numbers rising at a very rapid rate, forcing a shutdown in January. And around that same time, the vaccines were being rolled out. And they recognized that they didn't have enough supply to get the entire population vaccinated with the speed that they thought would be able to allow them to get ahead of the problem. So with that in mind, looking at the data, they felt that there was enough evidence that one dose could be protective enough, that their better policy to prevent, as Dr. Igor said, death and the severe disease was to vaccinate as many people with one dose. And they also feel that there's data that supports people getting the second dose even as late as 12 weeks later, that and it could still remain effective. So the recommendations of 3 weeks and 4 weeks is what it is, but there seems to be additional data that even as late as 12 weeks, people could get it and still be effective. Now, the counter to that is that if you start telling people they can get one dose and still be protected, and a lot of people might take that as an opportunity to only get one dose and never come back for the second. So it's obviously a trade-off, but the UK government felt that their own better public policy for now was to get as many people the first dose as possible. So that's what they're doing. They're still giving second doses, but they're giving as many people the first.
Yes, you know, the thing is, they're not saying that they only need one dose. They are just giving, they are trying to, you know, spread it, which was something that, you know, even the US considered, you know, at a point. And, you know, when you actually, I don't know whether it was a doctor or a Professor Fotoko who showed this today, or maybe in a previous presentation, you actually see how the antibody actually starts rising from the time you take the first shot, okay? And you will see that it is fairly very well sustained up to even 12 weeks and beyond. It is actually when you hit the, you know, it's like you start off your vehicle and then you turbocharge it. That's what really your second dose does. Your first dose will still be protective for a period of time. But we're not really talking about how sustained your immune response remains to be able to keep you at that level of not really getting severe disease and, you know, possibly dying. So, the J&J vaccine is coming out. Probably maybe that will be something that people will still think that, you know, just a one dose. But generally, I think, whether it's the UK, whether it's the US, the bottom line is that nobody has contended that the second dose is not needed. It might just be a question of, trying to, really get more people the first dose and give more time before they actually get the second dose, because the second dose is actually still needed, very much needed, at least for those ones, the mRNA vaccines.
Okay, see, John, I hope that helps. Getty, with your permission, we can pivot to the variants in the interest of time. We can continue to answer questions in the chat, but let's maybe touch on some of the questions pertaining to variants, which is obviously an issue of concern. So Dr. Igo, you mentioned that human antibody, monoclonal antibodies have been tested, and it's only, in your opinion, recommended for people with early disease. How does it affect the potential for variants? The monoclonal antibodies are just as they are monoclonal, and the variants are, by definition, going to deviate from there.
I'm glad you asked that question. That is a really great question. So first of all, there are many of we have two that have been approved for emergency use authorization. So the Lilly and the Regeneron. So the Regeneron product contains more than just one monoclonal antibody. So it's a combination of monoclonal antibodies. And so if I were to use a monoclonal antibody, I would go with the one that has more than one monoclonal antibody because of the issue of the variants that are coming up. It's likely to be effective against more variants because it has more than one product. So the Lilly approved monoclonal antibody, it's a one monoclonal antibody. But they are now trying a second product that has more than one product. And I think the preliminary result from that product is very, very good. It's effective in protecting people from against disease progression. And I think one of the message that we should take away from this call is that monoclonal antibody is available. It's being infused. And if you know anybody with COVID, at least in the US, make sure you call and ask for availability of monoclonal antibody. We are infusing an average of 8 to 10 every day in our clinic. It's being done in doctors' offices. It's an infusion. So it's being done, it can be done, it's all done in outpatient clinic. And the earlier you show, ideally we want people to come in within 4 days of their diagnosis. So the earlier you present, the more the likelihood that it will be effective. So the product is widely available and it's extremely very effective. And I think it's one of the ways to protect progression, and we should go for it.
Yeah, I think you also probably maybe speak to it's not every single person who tests positive that really will meet the criteria for infusion. So it's important that we don't all go away with the impression that everybody who tests positive should run to a doctor's office and get an infusion. So if you also can speak to, what are the requirements, what is it you're looking for in that makes somebody qualified to receive, a monoclonal antibody infusion.
I'm glad you raised that issue, Deji. So they're looking for people with high-risk individuals. So age and comorbidities are the two things. And there are criteria. So each of the clinic you go to would screen you, will pre-screen you whether you meet those criteria. And if you meet those criteria, they will bring you in. And out there in the community, it's working. And like I mentioned earlier on, I think almost everyone that we have infused have done well. But again, everything COVID, you never can say. All of them would have done well without the infusion. But we know in randomized controlled trial with hundreds and thousands of people, it's been proven to prevent disease progression.
Yeah, I just want to add to that. It's been our experience at my institution as well, that everyone who, and just like Dr. Deji and Dr. Ivo mentioned, it's really the high risk people who are at high risk of progressing to severe COVID, people at high risk of being hospitalized and dying, who it's indicated to use. And I've had the same experience as Dr. Ivo as well. Everyone that we've given the antibody infusions has not been hospitalized, at least knock on wood or something. And so we've had a good experience so far with that.
All right. I think we have, Aham, do you have any other questions?
Actually, Getty, if you don't mind commenting on the possibility of a Nigerian variant, that was something that came up early on in the chat. We saw a surge in cases in Nigeria in December, similar to seeing a surge in cases in Britain. Our data is probably not as granular as it should be, but nevertheless, anecdotally, it seems as if the mortality in Nigeria may have gone up recently. What are your general comments and thoughts on a possible Nigerian variant out there? How well characterized is it, and how impactful could it be?
From what I know, around the same time that South Africa was identifying a variant, they were identifying a variant in Nigeria that was actually different. And I believe that the Africa CDC was supposed to have been working on it with the Nigerian CDC. I have searched and searched to see if there had been further information. The last time there was any update was like the second week of January. They said they were trying to sequence it to be able to give us the particular areas of mutation on the virus. We're still waiting for that information. I certainly think that the newer variants had something to do with the second wave in Nigeria. And it's almost like our people are in denial. They keep saying there's no COVID here. We're not practicing as much social distancing or wearing of face masks or doing those things to help us mitigate. At the end of the day, we are doing this conference on vaccines, but the truth is that mitigation is still the good advice that we've been giving everybody. Wear your face mask, wash your hands frequently, maintain social distancing, and all the other stuff. People shouldn't forget that. And especially even after getting the vaccine, because of the potential of us still having our nasopharynx colonized by this virus. And even though we may be protected from the disease, we may not be protected from the infection. And we may be carriers and end up potentially infecting our loved ones and our friends. Until we get to herd immunity, we shouldn't stop. We shouldn't let our guards down, even if we're getting the vaccines. That would be my take-home message. But hopefully, maybe by the time we have another conference, we'll have more on the Nigerian variant. There is a Nigerian variant. They have not successfully characterized it as at this time, as far as I know. Thank you.
Okay, let me add, a little to what you've said. I don't truly know whether there's a Nigerian variant or not, whether it's actually speculative or not. One of the studies that we currently run with the National Institute of Allergy and Infectious Disease has a site in Nigeria, the study for the hyperimmune globulin, which is actually better than convalescent plasma, has a center in the Institute of Virology in Jos. And as late as today, Saturday, actually it's still this week, it's not last week, as late as, earlier this week, I was in contact with the Institute of Virology in Jos. They're actually supposed to be doing the sequencing of the quote unquote Nigerian variant, and still don't have anything. They say they cannot confirm the variant is actually what they think the Nigerian variant is any different from the ones that we've seen in South Africa, in Brazil, or wherever it is. So right now, there is no conclusive information from Nigeria that you actually, that you can pin anything, anything on really. So, but, and I actually, I want to actually raise, know whether we actually have anyone from Nigeria, from any of the government agencies or anything actually attending this conference today. Because everyone will remember that I was actually really very, very critical of the Nigerian government and the efforts that we were making. And we had actually put all our, all our eggs in one basket, relying totally on COVAX and not really going out to do vaccine procurement. And I'm sure that most of you must have heard the news within the last day or two about COVAX actually not giving the one promised 100,000 doses to Nigeria because they think Nigeria doesn't have the cold chain capacity to be able to keep the vaccines viable. So the 100,000 that we're supposed to be getting, 100,000 doses, which actually equates to 50,000 people that we're supposed to get in April, we're not getting it. Personally for me, it is devastating. It gives me concern every day. The 50,000 50,000 people that will be immunized with 100,000 doses, that doesn't even cover the frontline people in Lagos alone. So what are we doing?
It's been very grim, and with that in mind, it's a good segue to mention that AMPA is also organizing a similar discussion tomorrow pertaining to the management of COVID, particularly in the ICU setting. This discussion is going to be led by Gertie, as well as Yemi Johnson and Ngozi Ojioke, who's a pulmonologist here. So those of you who may have seen the flyer can join us tomorrow for that discussion, and it will probably give us an opportunity to go a little bit deeper into some of the areas Dr. Ofotokun talked about today, such as remdesivir, dexamethasone, and a little bit further deeper dive into treatments that we probably could have touched on today, but may have run out of time. So join us tomorrow. You can look out for that discussion. This is obviously an ongoing dialogue. My opinion is that it has been spectacular so far, and I would expect it will continue to be so. This is a rapidly evolving topic. There's new developments by the day, possibly even by the hour. So if we reconvene at whatever point the Infectious Disease Committee chooses, I'm sure we'll still have a lot of updating to do. But in the interest of time, we can put the brakes on it today and wind down. Dr. Anyanwoke, you can make some remarks.
Quickly before the remarks, this is actually, I just got information now that the information that I provided has actually been refuted directly from the WHO. So that's, I just wanted to make that correction before we close.
Okay, I see Dr. Okafor's hand up. As my Oga, the past president, we shall let him talk.
Okay. I guess the presidency has its privileges. Thank you very kindly, Dr. Anyanwoke. Just a little bit of commentary. I have to say, in my 25 plus years as an AMPA member, we've often struggled with what we call value proposition from this organization. I'll go on record and saying that this program here, Dr. Anyanwoke et al., everybody who's been involved in it has added so much value to this organization for me. I know there's the other financial aspect, but I can't commend you enough. Anytime I've been able to get away from call and join, I've come away much better. Thank you all very kindly. My main point on today's discussion is about monoclonal antibodies. And I go back there, not because of self-interest, because I'm over 60 and Black and male. Self-interest may be involved, but really on a very serious note, everywhere I've listened on this forum and other forum, it is very clear that giving early, especially if it's in short supply to a high-risk group, this can reduce morbidity, progression of disease, and mortality in effect. And I've been advised by the internist, oh, it's expensive, it's da, 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 da. Dr. Fosokon has it at Emory. But I'm here to tell you that many hospitals across the country do not have access to this. And I don't think the word is out enough that this can really change the trajectory, especially for a high-risk group. And the group here, talking here at AMPA, is speaking to that group largely. The Black and brown people over 65 with pre-existing conditions. And I'm wondering, I think I wrote earlier, as a matter of advocacy, maybe from us, more from NAACP and groups, need to begin to hear that really, if Black people are not getting to vaccines because of distribution, if they're not getting to vaccines because of resistance, if they do begin to fall ill and turn positive, there should be a general understanding and push to avail them of monoclonal antibodies. This is my central comment this afternoon. Again, thank you very kindly for letting me speak. Thank you.
Thank you, Dr. Okafor. You have just actually put an idea in my brain. I happen to know some NAACP people local here in Baton Rouge, Louisiana. And after this, I am going to get on the phone and see how we can do something together to work for the Black community. You have just put that in my brain. Thank you, sir. At this point, we've gone a little bit over our 4 o'clock mark. We really thank everyone that has shown up. Professor Fotokun, you are a jewel as usual. Dr. Adedeji Adefuye, my boss, thank you, sir. My dear friend, Obi Nedu, Chief Infectious Disease Fellowship at Oshna Medical Center, Fellow of the IDSA. That is really what you are. I am grateful truly and eternally. Everyone that has participated here, we are very grateful. I'm going to hand over to a member of our COVID-19 task force, Dr. Jide Bamiboye, to give us the vote of thanks. Thank you all.
Thank you, Gary. This again is an affirmation of the fact that AMPA is doing something really, really useful. And I want to echo Dr. Okafor's comment. Thank you all for attending this Zoom conference. Dr. Anyaoke is the only person that can gather a group of men together to attend a Zoom conference on Super Bowl weekend. So,
very true.
So I want to thank the AMPA national leadership, the current president, Dr. Chris Okunseri, attended and gave us a brief words of encouragement. Thank you for your leadership, and thank you, ex-Presido, Dr. Okafor, for the kind words as well. I want to thank Dr. Susan Ejonwe, Chair of AMPA National Membership, for our introduction, and the AMPA Secretariat for supporting us all the time. And I want to thank our speakers, the eminent scholar, Professor Igbo Ofotokun, Professor of Medicine in Infectious Disease at Emory University. Thank you for giving us very scholarly and eruditic talk on the vaccine. And also, I know that we've all been talking about ivermectin. Thank you for bringing it up. I wrote in the text yesterday that this issue will not go away regardless of what we think in the West. They will still continue to use it in Nigeria and India and wherever. Dr. Obi Nedu, thank you for your contribution always, Program Director of Infectious Disease Fellowship at Oshna. And Dr. Deji Adefuye, ever since we've been coming to Chicago to attend AMPA meetings, we've connected and you've always been very supportive of whatever AMPA does. And thank you very much in your new role as the Medical Director of Takeda Pharmaceutical. Hopefully you'll be able to give us in the brown and brown communities, making sure that we get Regeneron to give to our people.
I know your company doesn't manufacture it, but maybe you might have some leverage. Dr. Anyawoke, thank you again for your indefatigable work that you do, and we just appreciate you very much. You wear many hats. You are chair of AMPA COVID-19 Task Force, chair of AMPA National ID Committee, and the chair of AMPA Louisiana chapter. I don't know how you do all this, but you get them done, and you're very good at it. Thank you very much. And I also want to thank all the members of AMPA COVID-19 Task Force for their support, and the AMPA members in general for attending this Zoom meeting all the time that they call for them to attend. And also the general public. Mr. Diallo, thank you for attending. I'm still looking forward to visiting the first futuristic city, Akon, that is supposed to be the first in Africa, whenever that comes up. You need a lot of money. I hear it's going to be about $6 to $10 billion to set it up. Maybe half of that money can be used to buy vaccine. How about that? So thank you very much. Unfortunately, today we do not have a representative of the federal government. They should have been here to hear what we have to say and at least also tell us what is being done there in Nigeria. Hopefully they'll be able to join us next time.
Actually, Dr. Bangboye, we had Ijima Wankwo, who actually is a staff of NAFDAC.
Okay.
Who is actually on the call. And if you look at the chart, he actually provided some context around why what we were expecting from COVAXin in donor capacity might be delayed. But that is actually, it might be delayed, but not because of cold storage. So she's actually on the call.
All right, we'll thank her for attending. So we all know how Regeneron came about. It was in fact not available before, but became available after two prominent U.S. After the former president and his lawyer, got the Regeneron, and so they became prominent. So thank you all very much for coming. I hope that you will take all of this information that you have received here and take them to your community and kind of disseminate this information to them and let them know the importance of getting the vaccine. Thank you all very much, and may God bless you, and may your team win tomorrow. I am thinking that Tom Brady is my man. You may want Kansas City too, but I don't think so.
Not this time.
No, had enough.
Oh, no.
And thank you to Dr. Aham Onyike for his powerful moderating. It's been a wonderful run so far, and hopefully we'll continue doing this. Just having him as my co-moderator gives me confidence, and so I can troubleshoot behind the scene, knowing that it's in great hands, in the hands of Aham Onyike. Thank you, Dr. Onyike.
Yes, just sorry, it was, I didn't mean, it was not a slide. You are able, a very able moderator, and we thank you very, very much for that.
Thank you. Thank you all.
Okay.
Let's go Kansas City.
There you go.
No, Brady, if you are good, you're good. I mean, it doesn't matter if you win 6 times or 7 times or 8 times.
For everything, there is a season. Time for Brady to go. Time for Brady to go.
No,
for
this
everything,
is a
there
season.
is a season.
This is a season.
A time to come and a time to go.
Why does it really matter?
Bye, everybody.
Yes.
Bye.
Emmanuel, it doesn't matter how you all feel. He's winning.
We need to turn the leaf.
That's what you guys said when he left the Patriots.
We need renewal.
Yes.
We need renewal. A time for everything. Time
Don't
to
go
go.
Don't go back to Motorola.
Say, man, what are you doing? God, renewal.
All right.
All right. Nice one.
Thank you. Thank you.
Bye, everybody.
Bye.
Bye, everyone.
Bye, everybody.
Bye.
Bye. Nice lecture.
Talking amongst each other. So, it's no good anymore, right? Even if it was a group, right? Raising alarm, right? Government administration one and the anti with a group of doctors talking. Even in a mono something, even if prevent them from getting it, antibody